Okay, let's move on to our first speaker so that we take advantage of all the time we have. Our first speaker is Brooke Weigel from University of Chicago, and she's just finishing up her dissertation research. And Brooke, if you'd like to share your screen. Brooke's going to be speaking about differences in carbon cycling and microbial communities. And one of the great things about Brooke's work is she's working both on Tatoosh and at Squaxin Island in the southern terminus of Puget Sound. So there's a lot to add spatially to what we can learn about kelp. Thanks, Brooke. And I think you're muted, Brooke.
Yeah, sorry, one second. Having some difficulties for some reason. Okay. Can you guys see everything okay now? Great. All right, I will get started then. So I am Brooke, and I'd like to thank all of you guys for coming. It's so great to see so many faces and people who care about kelp. So today I'm going to talk a little bit about some of my research. First, I'll talk about kelp physiology and nitrogen requirements of bull kelp. Next, I'll talk about differences in carbon and nitrogen cycling between southern Puget Sound and outer coast bull kelp populations. And finally, I'll talk about the bull kelp microbiome and its functional importance. And so to begin, I'd like to acknowledge the Macaw tribe for giving me and my advisor, Kathy Pfister, permission to work on Tatoosh Island, and the Squaxin Island tribe for giving me permission to work with kelp from Squaxin, as well as Helen and the Washington State DNR for access. So I'm really interested in looking at bull kelp from the perspective of thinking about how energy and nutrients flow through ecosystems. Bull kelp are extremely large and productive primary producers. They grow multiple centimeters per day in coastal systems. And just reading the Puget Sound Kelp Conservation and Recovery Plan, it says that no information currently exists regarding optimal nutrient ranges for adult Nereocystis sporophytes. And I know that there's a few studies that have been conducted, especially with gametophytes, but we really don't know that much about the nitrogen requirements for bull kelp, which is surprising given that they are such large productive organisms. So I'm interested in looking at how much nitrogen bull kelp require and in what form: ammonium, nitrate, nitrite. And so I'll take you through some experiments that I did to quantify nutrient uptake rates by bull kelp blades. So in order to quantify changes in nutrient concentrations, you need a closed system. So I designed these tube chambers where you can pluck off one blade of the bull kelp, put it into this chamber filled with seawater, and sample the seawater over time to watch the nutrients disappear from the seawater as the kelp blades take it up. So in green, this is just an example graph showing changes in nitrate concentration over time in some of these chambers. I also accounted for the loss of nitrogen in the seawater due to the surrounding phytoplankton by using seawater control chambers. So I used this method. I also floated these chambers in a pool raft on the surface of the ocean, just below the surface, so that they stay cold and kind of rock with the waves and mimic the natural motion of kelp blades. And from many experiments conducted on Tatoosh Island, which is on the outer coast, I found that bull kelp blades assimilate nitrate at the highest rate. So this is a graph showing nutrient uptake rates by bull kelp blades. And they took up the most nitrate from the seawater. The mean uptake rate during the day was about 3.5 micromoles per gram of dry mass per hour. They also took up ammonium and phosphate, but at much lower levels. However, it's interesting to look at the preference versus the availability of these nitrogen sources, because in all of the experiments, the average availability of ammonium was only 2 micromoles per liter, whereas nitrate is much more available, and it's actually 9 times more available than ammonium. So the mean uptake rate of ammonium relative to its availability was actually higher than that of nitrate. So bull kelp blades likely prefer ammonium, but they take up a lot of nitrate from the seawater because that is what is available to them. And so next, I'll talk about some differences between kelp populations in Washington. So the nutrient regimes and nitrogen availability is really different between the outer coast of Washington and southern Puget Sound. Out on the outer coast, I work on Tatoosh Island, which is at the mouth of the strait on the Pacific Ocean. And the seawater dissolved in organic nitrogen concentrations are really high during the summer due to upwelling, some in the range of 15 to 25 micromoles per liter. Whereas in southern Puget Sound in the summertime, seawater nitrogen concentrations are really low, less than 10 micromoles, and sometimes in extreme cases, less than 1 micromole. And I'm sure Helen will talk more about this. And when you look at the kelp tissues between these two populations, you can see those differences reflected. So this is a graph showing the carbon to nitrogen ratio of bull kelp blade tissues from these two locations. And organisms have usually a lot more carbon in their tissues relative to nitrogen. But you can see on this graph that the blue circles are from those outer coast Tatoosh kelp, and they have a higher carbon and nitrogen content. And so overall, these kelp from Squaxin Island are, they're nitrogen depleted compared to these outer coast kelp. And so that is really interesting because it seems to reflect the availability of nitrogen in the seawater. So I wanted to ask, will seawater nitrate addition affect nutrient uptake and carbon fixation rates by these nitrogen poor kelp from Squaxin Island? So in pre-pandemic times, Helen Berry, Lauren Johnson, and Max Calloway helped to do this field experiment. It was a lot of fun. And basically we took bull kelp blades from Squaxin Island and used this chamber method to determine nutrient uptake rates. There were 2 nitrogen treatments: low nitrate, which was the ambient natural nitrate level at the time in the seawater, which was very low. It was 6 micromoles per liter. And then I added 30 micromoles to achieve a high nitrate treatment in these seawater chambers. And then I quantified carbon fixation rates using a stable isotope 13C tracer method, as well as nutrient uptake rates as I calculated before, using that chamber method of sampling seawater over time. I also calculated dissolved organic carbon release. And so what I found is that when you add nitrogen, bull kelp blades from Squaxin Island take up significantly more nitrogen. This is a graph showing dissolved inorganic nitrogen uptake. And in the high nitrate treatment, uptake rates averaged about 5.2 micromoles of nitrogen per gram of dry mass per hour. Whereas in the low nitrate treatments, nitrate uptake was only 1.28 micromoles. So when you give these kelp more nitrogen, they take it up. This isn't that surprising from kelp physiology. We know that when you add nutrients, they usually take up more nutrients, but I think it's important to look at, especially for these kelp in southern Puget Sound that are experiencing such low nitrogen during the summertime. I also measured carbon fixation rates. So on Tatoosh Island, carbon fixation by bull kelp is as high as 2.35 kilograms of carbon per meter squared of kelp forest per year. These are rates that we quantified and scaled up using the density of kelp on Tatoosh Island. But I also found that 16% of this carbon is released as dissolved organic carbon, or DOC. So this is a process that happens in all algae. As they fix carbon, some proportion of that carbon is released into the seawater as dissolved organic carbon, or DOC. And these are basically dissolved sugars and carbohydrates. And I like to think of it as analogous to just dumping tablespoons full of sugar into the seawater. I'm really interested in this dissolved organic carbon, and I'm happy to talk more about it with you. But I measured this carbon fixation and dissolved organic carbon release by bulk kelp from Tatoosh and Squaxin Islands. And as before, I found that these southern Puget Sound kelp have significantly less nitrogen in their tissues, and bulk kelp from Tatoosh Island fixed significantly more carbon and produced more dissolved organic carbon than kelp from Squaxin Island. I will say that this was with limited replication at Squaxin Island. I did a lot of experiments at Tatoosh over multiple summers, but only one big experiment at Squaxin. So we need to do a lot more measurements to follow up on this trend of differences in productivity. Finally, I'll talk a bit about the bull kelp microbiome. I'm interested in asking the question, can knowledge of the kelp microbiome inform kelp forest restoration efforts? But first, we need to understand the attributes of healthy kelp microbiomes, which includes understanding the functional role of dominant microbial taxa in order to understand how the kelp microbiome might affect the health and productivity of kelp forests. So what does a healthy kelp microbiome even look like, especially for the bull kelp? To begin, I sampled bull kelp microbes across the state of Washington, and I found that they display geographic variation. So I sampled bull kelp blades from 13 locations from southern Puget Sound through the strait and to the outer coast. And I did 16S microbial gene sequencing to get a picture of the microbial communities. And you can see from this gradient of colors that the microbes varied across sites, but there are core taxa that are always present and are possibly important members of the kelp microbiome. I collaborated with Drs. Tabitha Ramirez-Puebla and Jessica Mark Welsh at the Marine Biological Lab to use a novel imaging technique that reveals the micron scale spatial structure of the microbes. So it basically allows us to visualize what these microbes actually look like based on the DNA sequences of the microbes that are present in the community. And using this technique, we found that bull kelp blades host a dense microbial biofilm. This is a sample collected from the tip of bull kelp blades on Tatoosh Island in the summertime. And you can see that it's just covered with bacteria of different species of bacteria, and it's pretty dense. So I wanted to know if there's differences in microbial communities between the outer coast and these kelp in southern Puget Sound, which Helen will talk more about, I'm sure, today. But we know that the kelp at Squaxin Island have been declining and are relatively unhealthy compared to the kelp at Tatoosh Island. And using this imaging technique, we actually found that the kelp from southern Puget Sound have drastically lower microbial cell densities than kelp from Tatoosh Island. So this is an image, it's a bit more zoomed out than the previous one I showed, but you can see that there's tons of microbes covering the surfaces of kelp from Tatoosh, but kelp from Squaxin Island had very few bacterial cells. The community was really sparse, but these images were collected in 2018, and we wanted to know if this trend persists across time. So we sampled the next summer in 2019. And again, this graph on the right shows a different summer, the same trend, that there's many more microbes at Tatoosh Island compared to kelp from Squaxin Island. So you were probably wondering, but what is the importance of the kelp microbiome? What are these bacteria even doing on the kelp? So we now know that there's more than 25 million bacterial cells per centimeter squared of kelp tissue. And we all know that bull kelp have really long, beautiful blades. So that means there's trillions of bacteria living in these kelp forests. And I think it's important to understand what they're doing, especially because they have such a high abundance in this ecosystem. So I'm not going to get into the details, but I used metagenomic sequencing to sequence all the DNA present on the kelp blade. And I assembled 80 bacterial genomes from kelp blades collected at Tatoosh and Squaxin Islands. And I found that for the most abundant bacteria on bull kelp, these are these little pink circular cells called granulococcus, which there's more than 1 million cells of this genus alone per centimeter squared of kelp. I found that in their genomes, they have a lot of genes to take up carbon or sugars that are released by the kelp. So they have all these transporters that are bringing carbon into the bacterial cell. And I also found that in their genomes, there's a lot of genes for synthesizing vitamin B12, which is an important vitamin that the kelp likely need and probably don't synthesize themselves, although we need a full genome to figure that out. I also found genes that the bacteria are reducing nitrate to ammonium, and they also hydrolyze urea into ammonium. So it's likely that these bacteria are providing bull kelp with a source of vitamin B12, as well as another source of ammonium, which seems to be a preferred nitrogen source for the host kelp. I know I'm basically out of time, but conclusions and next steps, I think it's important to continue looking at how nitrogen impacts the physiology of bull kelp. What is the threshold limit that they require? And how does low nitrogen interact with other stressors like temperature? The bull kelp microbiome is providing kelp with a source of vitamins and nitrogen, and the southern Puget Sound kelp have a lower microbial density. So I think it's important to look at the causes of this low microbial density. And I'd like to acknowledge my advisor, Kathy Pfister, and everyone else who helped make this research happen. Thank you.
Thank you so much, Brooke. I see people applauding, and you're getting some comments in the chat. And I'd also like to open this up. We have about 5 minutes. We have a couple of minutes for 1 or 2 questions, if anyone has a question. And go ahead, Gray. You have your hand raised. Oh, I see you were clapping. I have a question that I think a lot about for Brooke, which is obviously microbial communities are incredibly important to understanding the health of the kelp beds. But what I'm really interested in, and I don't know if you have more thoughts on, is how we link that to many of the on the ground projects that are going on within this group. So if you have any thoughts on that now, it would be great. And then also you've got another question, you get to decide which to answer, from Margot Hessing-Lewis. Do you have any information on the annual succession of the microbial communities?
Yeah, I guess to begin, we do have information on the annual succession, and they do increase in abundance and diversity over the summer growing season. And we have evidence that these microbial communities are forming on like new kelp blade tissues and then growing as the kelp blade elongates. And I can put my paper in the chat that shows that. And then Helen, you were asking how how the knowledge of microbial communities can be integrated to some of the other efforts that are ongoing. Is that what you were saying? Yeah, I mean, I think we probably need some more sampling, especially in southern Puget Sound, to really understand if these kelp actually have lower microbes, because we have samples from 2 summers, but it would be great, to know for sure that this trend is real. But we also don't know if the low microbial abundance is a consequence of kelp being stressed, or if it could potentially be a cause, or if it's related to their health. I'm guessing it's a consequence, but again, I'm finding that these microbes might be important if they're providing vitamins and nitrogen to the kelp. So I just think we need some more research in order to determine the exact causality and direction of this interaction.
You have time for one last question that you got, which is a really fun one from Tish, which is, great talk, Brooke. I'm curious about whether there could be a connection between the microbial community and some kind of protective function against fouling organisms such as bryozoans.
Yeah, that's a good point. Someone actually just published a study showing different microbial communities on macrocystis that were fouled by bryozoans and ones that were not. Bacteria in general tend to produce antimicrobials, and they tend to structure their own community. And I've done studies that show that those granulococcus are the first to arrive. They arrive on the bull kelp tissues within a few days. And they also, I found out from their genomes that they have flagella and chemotaxis genes. So those bacteria are probably swimming towards the kelp and landing there and establishing. And so they might be preventing other organisms from arriving, but I'm guessing the kelp itself is regulating some of those things too. I don't imagine that. I don't know if the microbes can prevent bryozoans from settling or not. That's an interesting question.
Okay, we're going to wrap this up, and we're out of time. Thank you so much, Brooke. You have a few more questions in the chat. Perhaps you could answer those. And what we'll do now is move to the next talk by Eliza Heery, who some of you may know from her graduate work here years ago. And she's going to tell us about a new network of monitoring that they are instituting.
There we go. Can you hear me?
Yes.
Great. Well, thanks so much. And I really want to send a big thanks to Helen and everyone at DNR for setting this up. I feel like it's just really important and something that we really need to be doing, all talking. So I'm a postdoc with Marine Geo, which is a group within the Smithsonian. I'm based at the Friday Harbor Labs. So I wanted to tell you a little bit about what we're doing up here relating to kelp work. So first off, what is Marine Geo? It's a network of scientists led by Smithsonian that coordinates annual monitoring across network partners, which are all over the world. And traditionally, the focal habitats of Marine Geo have been seagrass, coral reefs, but there are a lot of other habitat types that are included in Marine Geo sampling at different network partner sites. So I think this is still true that Friday Harbor Labs is Marine Geo's newest network partner. We joined several years back. And starting off, we have two focal habitats, eelgrass beds and kelp beds. So eelgrass sampling, of course, Drew Harvell's lab has been doing eelgrass work here for a long time, as well as other Sandy, Wiley Echeverria, and even others before that. So there's a long history of eelgrass monitoring, and much of getting that part of the Marine Geo FHL monitoring system up and running was really just partnering with Drew's group. So that started in 2020, the summer of last year. And kelp bed sampling is something we want to start this coming summer. So one of the core goals of Marine Geo is to develop protocols that are publicly available and standardized, that can be used both across the partner sites within Marine Geo and by others. So it's everything that Marine Geo is developing in the way of protocols is available on their GitHub site. Their standardized protocols are organized into modules, and a lot of these are things that can be implemented across different habitat types. But then on top of that, there are protocols that are kind of specialized for focal habitats. So for seagrass beds, for instance, you've got prioritized, so our core modules, these four, and then you have these other recommended modules. So that's kind of just an overview of how they're typically set up. And then each of them, each of the modules ties into global ocean observing systems essential variables. So the Kelp protocol for Marine Geo is currently in development. So the protocol development process is already kind of fixed. Marine Geo follows a standard workflow in how protocol development, testing, and revision works. That's all outlined. Is there a way, Helen, to make the slides available later for the links? I can also just provide the link separately.
I'm happy to post any materials on the website. So yes.
Okay, cool. So the workflow for protocol development is all posted on the GitHub site as well. Basically, it's coordinated by this internal working group, human working group, and then it's carried out by a protocol lead, which is one or more of the network partners. So for the Kelp protocol development, we're trying to kind of lead the way, and Hakai has been really, Margot and her group, glad to see Margot on the call, been super helpful in helping us get this together. So the steps involved are kind of as follows. First outlined there is to collect and compare community accepted protocols and to assemble kind of comparison tables, just make sure we're coming with an informed perspective of what has been done, what is typically done, and really tease apart from that whether there is a need and how we can fit into it. So that is something we were pursuing for much of last year, at least this summer when we weren't able to dive. This is a kind of snapshot of a portion of that table, which is a Google sheet, and I'm happy for that to be shared too. It's a bit longer than this, but compares all the different major protocols that are out there in a variety of different metrics. So based on that, and we identify where additions, subtractions, modifications might be needed to fit marine geo's needs and to maximize kind of efficiency and the value of data with specific goals in mind for what questions we can ultimately answer, and making sure that they align with the essential ocean variables. Those are kind of the criteria for that next step of drafting the proposal, sorry, the protocol. So this is where we have been leading up until kind of last few months. We've got some things drafted, but we now really need to kind of a workshop, which I'm again glad to see Marcus on the call because we need to find a time to do this. But I think later this month, we're looking to just kind of get a working group together that is going to kind of finalize different trade-offs and feasibility of all the different alternative methodological details, kind of small nitty-gritty details, really. Sorry, I should have that muted. And then from that, we will be drafting the kind of final version. And I include here that others are definitely welcome. We are free to email me if you're interested in joining for nitty-gritty protocol details. So whatever is finalized there, and I'll circle back to this in a minute in terms of the details that are likely and what we have drafted thus far, but whatever does come out of that process goes through an internal review within this working group that we're part of, and then an external review with the Marine Geo Network, with the data working group, which is separate within Marine Geo, and then with external topic experts, which really will include, I hope, all of you. So thank you in advance. We'd love any and all feedback you can provide at that time. And then it's a process of revising and testing all of the protocol details that are in there, integrating reviewer changes, and also each protocol module comes with field sheets and data entry sheets so that it can be pretty easily updated or integrated into the Marine Geo's database system. There will then be a period of just finalizing all of this and getting it up on the website. So that is kind of the overview of how protocol development works. So because we're kind of in the midst of that process, I don't have the final version of it to give you. I was actually hoping we'd be further along than we are, but I think I can share what seems likely from the process thus far. So it does, I think we're probably looking at using fixed or semi-fixed transects. So probably with fixed start and endpoints, possibly more involved in that with a kind of more permanent transects along the entire thing. I think it would probably just be markers. 30 meters of transects is most likely because that we're trying to tie this in with all the different Go Keen, all these different groups that are out there, as well as the Goose macroalgal working group. So that's probably what we're looking at in terms of transect length. Again, a lot of it is up in the air. There likely will be some sort of threshold sampling as well. I hate to be so wishy-washy about details. I guess for macroalgal sampling, I can say we're trying to tie pretty strongly into the Goose macroalgal working group protocols that are also in development. I don't yet think they're public, but from what we understand, the taxonomic resolution used there would be kelps, parrot macroalgae, and invasives identified to species, and then the Katami functional groups used for all other macroalgae. Likely the components will include would be uniform point counts, also swath sampling with probably a 2-meter width, so getting population density of macroalgae, mean thallus length, other morphological traits. And then also, I think getting size structure information for grazers is going to be really an important piece of this as well. We are a little bit restricted in terms of the parameters for visual surveys because Marine Geo follows a protocol that basically is the reef life survey protocol. So they use 50-meter transects, which is among the different trade-offs we need to work out for the final draft of all of this. The visual surveys and water quality protocols are already pretty much there and available online. So that's what will be adopted for kelp bed habitats as well. And then each of these modules also, I'm thinking they'll probably be optional. The only one of these that's not already part of the protocols for Marine Geo generally is the kelp morphometrics. But again, much more in the way of details to come there. So the goal here is really to generate data that are globally standardized for one, also regionally and locally really relevant. They will be public and should be informative and useful for as many people as possible. So if there's any part of that that is of interest or you just want to chat more, do just go ahead and reach out. I'm happy to hear from everyone and anyone. I wanted to add a brief aside just about.
So Liza, we've got to wrap up. Thanks.
Okay. Sorry, last slide. A brief aside about my, I'll just run all these out. My kelp related work. One is on endophytic kelp communophytes. This is my research that's separate from Marine Geo. I'm finding that endophytic kelp communophytes are really common in red filamentous turf algae in heavily urban sites in Seattle. Also that kelp, understory kelps are less abundant and they have a lower canopy height in urban areas. So if anyone wants to talk about urbanization, I'm also around and would love to hear from you. Many people to thank, and that's just a quick slide there, but other than that, hopefully there is maybe time for questions.
I'm sorry to say, Eliza, there isn't time for questions,
Okay,
but
sorry.
what you could do, it's great. Thank you so much. And what Brooke is doing and you can do now is people can add their questions and connections in the chat, and then we can keep that conversation going. So there's plenty of time for questions and discussion. And to keep us on schedule, I'm going to move to the next talk, which is, hold on one second. I'm hoping you all could see a single slide on changes in bull kelp over 145 years. This is
no screen sharing yet, Helen.
Okay. Let's see. Thank you for letting me know. Can you see it now?
Yep, we can see it now.
Great. Thank you. So I'm going to give a really quick set of highlights over a paper that came out a couple of months ago, looking at changes of bull kelp over 145 years in South Puget Sound. The authors are the Nearshore Habitat Program gang at DNR, including Emeritus Tom Mumford, And then I want to highlight some other people who are involved from USGS, Eric Grossman and Nate, and some data contributors. And I'll talk about the important data they contributed. Tarang from PNNL and Christopher Krems and Julia Bosch from Ecology. And also Ron Thom was really important in providing us with background for this work. In a nutshell, our objectives were to describe changes over time to historical extent of bull kelp in South Puget Sound. It's an area that we're concerned about from observations, and it's also like a canary in the coal mine, being the southern terminus of Puget Sound. And the main goal that I'll talk about today really is to place the results that we got in the context of regional data about different conditions so that we can all draw more inferences about stressors and put some of this thinking into our future work. So here's what we did. We synthesized all the historical data we could find because there isn't a lot of standard scientific data on bull kelp distribution. We found sources from 1873 to 2018, over 3,000 observations at the scale of 1 kilometer segments. And then we recorded either present or absent bull kelp on the -6 meter bathymetry line. And you can see a little picture there, an 1873 map. You can see little wavy symbols. That's the kelp. Pink means present, blue means absent. And then we analyzed the data, as I said, along 1 kilometer shoreline segments, and it was 450 kilometers of shoreline total. In a nutshell, here's what we found. Here's a map of South Puget Sound. The pink areas are areas where kelp has been found most recently, and the blue is the cumulative total. So the recent distribution is 80% less than the cumulative total over the study period. And we were able to establish a baseline from the 1878 period, and we found the current distribution is 63% less than that first historical baseline. So there's been a lot of loss, which doesn't surprise us. And pretty much bull kelp is common around the Tacoma Narrows now. You can see the pink around the Tacoma Narrows. And then this innermost bed that Brooke talked about, and I think many of people are fascinated by, which is the Squaxin bed, which is the southernmost bed or the most innermost bed that we've ever observed in Puget Sound. So you can drill into that last map a little more and look at temporal and spatial patterns. So this map shows the most recent time that bull kelp was observed at a 1 kilometer segment. And purple is a long time ago. You can see time was binned into 20-year increments. So deep purple is 1860 to 1880. Going up to, since 2000, is deep green. And what's clear is that bull kelp losses began early and then continued throughout the entire time period. And losses were concentrated in what we're calling the west and central subbasins, which are to the west on the map and outlined roughly at the top of the map. So that's the pattern, and this wasn't surprising to many of us. We had suspected this. I am going to go a little bit into why for this group, because this is really the question that we need to figure out now. And as I said, all we're doing is synthesizing data and making some basic inferences in this study. First, I want to highlight a hypothesis by Sam Starko and Chris Newfield, and Chris will be talking next, that wave-sheltered shorelines are common in the Northeast Pacific, and they may be especially sensitive to kelp losses. And this is a paper that that they published in PLOS One in 2019 that looked at the distribution of sheltered shorelines in the Northeast Pacific and made this point. And the take-home message is that if kelp along sheltered shorelines is more sensitive, that would be very important for us to structure our research and response around. And then finally, I'm not sure what the mechanisms related to this sheltered versus exposed differentiation is, but we all, I think, are thinking a lot about what are the temperature and nutrient conditions in these areas. So we closed out the paper looking at some patterns, first in wave height versus daily currents. And that left plot, what you'll see is the x-axis is wave height from USGS. And this is current wave height. We don't have historical data, but it probably hasn't changed hugely. And the y-axis is maximum daily currents. And what you'll see is that the deep green squares are the area where, or the deep green are the areas where kelp persists in recent decades. And you'll see that most of them are above 1 meter per second daily currents, mean maximum daily currents, which suggests that that currents and mixing, really strong currents and mixing, especially around sills, might be playing a role in resilience once you get inside these generally wave protected environments. The x-axis you'll see is looking at annual maximum wave height, and there's not a clear pattern in kelp persistence. And then the last thing I want to mention is temperature nutrients. We used mid-channel long-term, so this is multi-decadal data from the Department of Ecology, which we greatly appreciate. And we found pronounced seasonal and spatial patterns. Specifically, values are similar in the dark blue, which is the narrows, and the deep red, and places in between. Deep red is squawks, and values are similar in the winter, and then they diverge strongly in the summer. And the divergence more extreme the farther away you get from the Tacoma Narrows. And of course, high summer temperatures are accompanied by low nutrients. And we saw these same patterns in nearshore data that we collected between 2017 and 2018. And so we felt that we could draw conclusions from the broad patterns from the mid-channel stations. One of the questions I wonder a lot about is, what are the thresholds for damage or impact to species like Nereocystis related to temperature and nutrients? And Brooke talked a bit about this. This is something that I think that we're really looking to the research community to help us to understand better. Are we hitting single or combined thresholds? And is this impacting what we're seeing in the distribution of kelp? And luckily, I think Robin Fales might be on the call. She just got fellowship funding, and hopefully she can add a little detail of it into the chat. And she'll be looking at temperature and nutrient levels in some work at Friday Harbor Labs coming up this summer. So on that note, I'm going to stop sharing. And I'm not sure, let's see, we have time for one. I'm going to move on because we're pretty tight time-wise, but anyone can ask any questions in the chat. And on that note, I'm going to hand the next presentation to Chris Newfield and Sam Starko to talk more about what they've learned on the in the Banfield area and how it informs our work here.
Great, thank you, Helen. Can everybody hear me all right? Yes, good, okay, and you can see my screen. So we'll try to make this quick. And actually, Sam and I have been co-teaching together for many years in Banfield, and so we're gonna try to make this a 10-minute tag team conversation. So I'll start and I'll pass it over to Sam, and I can be the one to advance the slides as we go. So I guess first off, thank you so much for the invitation to be here and to talk with this group. It's really exciting to see so much wonderful work going on, so much collaboration, and really, you know, just especially today, I can just see so many ways in which the work that we're all doing really connects well to each other and can really kind of collectively be a lot more than what we each do individually. So I'm excited to be part of that research community. So today we're going to talk. So rather than kind of thinking and following from what what Helen just said, we're going to kind of think a lot more about what some of those local drivers of kelp forest resilience can be. Before we get started, I think I'll just quickly acknowledge a number of important collaborators on this. So Julia Baum is Sam's postdoc advisor at UVic. Louis Druhl has been working on this stuff out of Banfield for many, many years, and we've relied heavily on his historical data sets collected over the years in Banfield. Leanna Gendel and Brian Timmer have helped in a number of ways, including on some ROV surveys that we'll talk about. And we've also benefited a lot from a collaboration with Jen Yakimishin and others at Parks Canada as well to access some of their data. And then finally, A lot of this work started sort of off the side of our desks as we were teaching in Banfield, and so we have many, many undergraduate students to thank, but in particular these listed here who helped collect some of the initial kelp data from some of these photos we'll show. Okay, so we know that kelps are sensitive to marine heat waves. This is from a study published in 2016 by Tom Swarnberg and others, showing the changes in kelp forest and really replacement of kelp forest habitats by seaweed turfs following the 2011 marine heat wave in Western Australia. And right around the same time that that study came out, as we all know, we experienced a similar marine heat wave in our part of the world. And that heat wave had direct effects on things like sea surface temperature anomaly, as shown on the left here, but it also had indirect effects through exacerbation of sea star wasting syndrome and cascading effects throughout the ecosystem on urchins, et cetera. So there's important impacts, both direct and indirect, from these marine heat waves. And so this is a map that shows studies on the effect of that marine heat wave I just showed on kelp that have been published since about 2016. And they're coded by sort of what their main outcomes were. So blue shows studies that found resilient or persistent kelp populations in the face of these heat wave effects. Orange shows locally variable effects, and red are studies that predominantly showed negative impacts of that marine heat wave. And so one thing to draw from this is that it's clear that these heat waves aren't just affecting species at the warmer edges of their ranges, and that these heat waves have spatially variable impacts, especially looking at the distribution of those orange studies through these sites, that are really challenging to explain by just looking at these regional averages. And so we can kind of miss the local scale impacts of these temperature events if we only think about the regional scale drivers. And this is a point that was made by Amanda Bates and others in this 2018 perspective piece, just saying, we're ignoring these kind of localized ocean weather events at our peril, that we really need to think about these localized scale factors to really think about how ecosystems like kelp forests are going to respond to these kinds of climate events, which we know are becoming more and more common. All right, so I'll pass it over to Sam to carry on.
So as Chris and Helen mentioned, we've been working out in Barkley Sound, which is on the west coast of Vancouver Island. So not right in the Salish Sea, but pretty close. And what I think is really interesting about Barkley Sound and why we think that our system is relevant to really other parts of British Columbia, including Puget Sound, is that we have all of this really complex environmental variation that arises through the complexity of the coastline and the positioning of the islands throughout the sound. We also have some really, really interesting environmental gradients that I think really closely parallel those that we see in the Salish Sea. And so I just think, or we really think that this is a perfect study system to start thinking about general questions about how those local scale, how that local scale in environmental variation, or sort of quote unquote ocean weather, might impact the resilience of kelp ecosystems. And so this has really motivated a lot of our research recently, trying to really understand what factors influence the vulnerability versus the resilience of kelp forests to the direct and indirect effects of warming and marine heat wave. And so in a very similar fashion to the project that Helen just presented, we've also been working in this place that has this awesome historical this awesome set of historical records dating back quite a ways. In our case, not going quite as far back, but dating back to about the 1970s. And so using images and site level field data collected in the field through boat surveys, maps, as well as aerial imagery, we've sort of just been piecing together the historical distribution of kelp forests, both intertidally and subtidally across Barkley Sound, to try and ask this question. Next slide. And so we've started to note, we actually, right up during this heat wave, just being out there and working in Barkley Sound, we really did anecdotally start to notice some differences, both below the tide line, but also just really in your face in the intertidal, such as this photo shown here of Wizard Islet, where we can see this shift from this brown algal dominated community in the intertidal of rockweed and kelps to this sort of turfier green algal dominated community. And so I won't talk too much about this, but I just want to give a shout out to another shout out to our paper published in PLOS One, where we actually looked at the impacts of, or I looked at changes in intertidal communities, comparing sort of recent surveys following this marine heat wave to historical data from the 1990s. And what we actually found is that there's been declines across a lot of Barkleys Sound. And we looked at a number of different predictors, but really the most important, or what seemed to be one of the most important drivers, was wave action. So again, this is pointing to a possible role of local mixing in impacting kelp forest resilience, at least in the intertidal zone. Next slide. And so this pattern was largely driven by changes in the distribution of some cosmopolitan species like Alaria marginata that was previously found. So these are just showing bar plots showing an abundance category of Alaria across different sites on the top. And we can see that in between 1995 and our resurveys in 2017, 2018, we really didn't see much change at exposed sites, but at moderate and wave sheltered sites, Alaria did experience some precipitous declines. However, we didn't actually see this same pattern across all of their species, suggesting that different species might be affected by different environmental variables or might be impacted at different scales. And so a really good example of this is shown with Macrocystis at the bottom, or the giant kelp, where although we did see some really significant declines across time periods, there was no interaction with wave exposure, which suggests that something else was really driving these patterns.
Okay, so this is just kind of moving from that intertidal study, which really, in a lot of ways, I mean, we looked at wave action. The mechanism we think was happening in the intertidal is that wave action was directly having a cooling effect on intertidal macroalgae, and so sites that were wave exposed and wave sheltered were distributed at a very fine scale throughout the sound, so both closer to the mouth of the sound, but also further in. But as we started looking at species like Macrocystis, we also realized and now have good data to show that as one moves further into the sound, from these outer coast sites here to these kind of more inner coast sites here, there's about a 3 degrees Celsius predictable increase in temperature, and some of these intercoast sites are experiencing temperatures up to almost 20 degrees Celsius in the warm summer months. And so it turns out that gradient is really important it seems to be really important in driving the losses in kelp that I'm going to show you as we move forward here in subtidal kelp. So this is a slide just summarizing, again, our study region, but sort of 2 key takeaways here. So you don't need to know all the details, but the first thing that I'll point you to is that, so we had a whole bunch of point locations where we looked at presence or absence of kelp beds over time for these subtidal sites. That's shown in the blue and green points here. And then we also had, we kind of focused on this region near Bamfield from Cape Beale all the way in further into the sound, where we had better historical aerial photos. And that's what I'm going to focus a little bit more on going forward. So this is an example of an aerial photo from a shore zone flyover that was done in that region in 2007. And basically what we did for that whole region is that we divided the coastline into these 30-meter segments based on known landmarks that we could see from the air, but also that we could see from the water. And so this is just an example of a particular shoreline near Bluestone Point, near Bamfield, where you can see these different colored lines here as individual shoreline segments. So most of them are quite short and easy to quantify. And then when we got to these places like open bays, a few of them were quite large, but also places where kelp was largely absent in these sandy habitats. Okay, and here's what we found. So quite strikingly, we found that over this large time period from before till after the marine heat wave, kelps have disappeared from about 43% of the shoreline segments. And most of the losses were distributed at the inshore sites. So this is kind of our study region near Bamfield here, turned on its side. This is the open coast over here. Further in over here, and blue is coded as kelp forest sites being persistent over that time between 2007 and 2018. Red is coded for kelp sites or shoreline segments where kelp was subtidal kelp was lost. And then on the bottom here, we've actually separated that out by species, so we can see the probability of extirpation at any particular site and across sites as a function of distance from the open coast. So as you move from Cape Beale further into the sound, you can see that the probability of extirpation for macro increases dramatically. Same for Nereocystis, and then this third plot is just showing the same pattern for both species combined at these sites up in here. So quite dramatic changes over that time. And the other thing we found that these losses have persisted over time. So similar to that Wernberg study that I showed, these changes in kelp at this local scale have persisted since the 2014 to 16 marine heat wave. And so they've continued throughout these regions, especially in these wave sheltered habitats. Except we also know that there's a few sites quite far into the sound where the kelp has actually persisted. And so we wondered if those could actually be acting as refugia, and wondered if there was something different about those locations that might have helped kelp to persist in those regions. So that's what led us to this next set of studies that Sam will talk about quickly.
Yeah, very quickly. So then really to try and understand why some of these kelp forests were persisting in these locally warm areas, we conducted a series of these ROV surveys with this mini underwater drone, where we would just run transects at 35 random points along that gradient in towards shore. And we looked at the substrate and whether and basically how deep the kelp went. And then we also wanted to look at urchins, because we had this hypothesis that perhaps if urchins were setting the lower limit of kelps, that might prevent kelps from inhabiting deeper waters that might actually act as thermal refugia. So we did these ROV surveys, and what we found is that a lot of these refugial inshore sites were actually found on sort of sandier habitats that tended to lack urchins. So the black line is just showing, on the top plot is just showing that the probability of kelp presence decreases as you move into the sound, which is the pattern that we saw generally in all of our other surveys. However, at these sandy sites, we actually started to see that whereas up towards the outer shore, these were sort of exposed sandy beaches that were absent of kelp, they tended to be quite good kelp habitat as you moved further inshore. And this is really because these beds were able to get much deeper below the thermocline into much cooler and more nutrient-rich habitats. So I'll just walk you through a few examples of what these look like. So towards the outer shore, what we found is that the kelp beds really didn't go that deep, and they were really just found in this fringing area between between the intertidal and just a couple meters below, where they would then run into these urchin barrens. However, similar types of habitats, so rocky habitats, towards the warmer edge of our gradient, we still found that macroalgae was extending to several meters below the intertidal, but now there was no kelp at all. Instead, what was going on was this sort of macroalgal turfy reef meeting with urchins. So this really suggests that the shallow waters are not habitable for kelps at these inshore areas. And then lastly, we found that some of these refugial habitats were actually really deep. And so these would extend to depths of 12 to 14 meters, so that they were well below the thermocline in really cool areas. And what's really interesting, and we don't have time to get into this too much, is that some of these, a lot of these were actually these Macrocystis populations that were lying totally flat on the substrate at depth. And it wasn't until sort of their shallowest edge, which was maybe at about 5 meters, that you would start to see them reach up towards the surface. And so these beds were really much more expansive at depth than what you were able to see from the surface. And so just to sort of wrap all of this up, we found that local environmental variation can play a really important role in driving the responses of kelps to large-scale warming events. We also found that the impacts might differ between species, and that I think we really need to think about things like microclimate, such as local environmental variation, such as microclimate, as something that can lead to vulnerabilities really far from species range edges. And I think this is especially true in basins such as the Salish Sea or such as Barkley Sound that might have less water exchange. And finally, just a last point is that any potential changes in the environment really have to be thought of in the context of other drivers, such as changes in trophic dynamics, because these things might interact to really result in a patchwork of remnant ecosystems. And with that, can put our contact information up here. And I don't think we have any time for questions, but be happy to take them in the chat.
Thank you so much, Sam and Chris. Yeah, we've got a great conversation going on in the chat. And so I'm hoping everyone will add questions for Sam and Chris in the chat. And I'd also like to thank them for starting to talk about urchins. The next discussion will be a 3-part presentation followed by some discussion starting with Taylor Frierson at WDF&W about some initial observations of increased urchin populations on the strait.
Yeah, hi everybody. Thanks for having me. Can you hear me okay? Okay.
We can hear you fine. Are you sharing your screen yet?
No, I will do that right now. Is that working?
Looks great.
All right, cool. So yeah, I'm Taylor Frierson. I'm a subtitle shellfish biologist with the Washington Department of Fish and Wildlife. I work with Hank Carson, who I think would normally be joining this meeting, but he's underwater today. So I am the fishery manager for the sea urchin and sea cucumber fisheries in Washington. And yeah, I'd like to give you a preliminary assessment of purple sea urchin grazing on kelp communities in the Strait of Juan de Fuca. Yeah, so a little background on purple urchins in Washington. They are relatively common out in the Olympic Coast National Marine Sanctuary in the Strait of Juan de Fuca and the San Juan Islands, but their abundance is not nearly as great as we see with the red sea urchin and the green sea urchins out in the same areas. So they've kind of been not really really observed or considered much in kind of Washington's past. There is no commercial harvest of purple urchins, although we do have a substantial red and green sea urchin fishery. But there is a small scale recreational or personal use harvest that occurs in Washington. We estimate maybe 1,500 total are harvested every year, and that's from a harvest survey that we were able to do. So really, there's minimal data that exists for purple urchin in Washington. Again, we with our red and green urchin fisheries, we conduct surveys every year for stock assessment and other indexation purposes, but really not looking at purples at all, and really don't see many on our surveys in the places that we're going. So very little data. You know, as recently as like 2018, we were getting phone calls and pictures from sport divers sending us pictures like this one on the upper right here. This is Tatoosh Island from 2018 or 2019. So pretty stunning picture. You see the densities of purple urchins actively grazing on Pterogophora, and then a figure here shared from Ole Shelton. The NOAA dive team has been conducting surveys out in the Olympic Coast National Marine Sanctuary for some time, and they did count purple urchins. And you'll see really quickly, Tatoosh Island as the blue line, very rapidly increasing densities since 2016, 2017. And we're looking at two different depth bins here. So the upper one's 5 meters deep, the lower one's 10 meters deep. So yeah, it's gonna put those together, and we had some concerns or questions what something could be happening out in that area. Let's go have a look. And I'll take a couple steps back here. Why are we even talking about purple urchins And I think everyone here probably knows this story pretty well by now. California urchin and kelp paradigm. I'll just kind of start with Central California, which I think is maybe a little more similar to what you might be seeing here in Washington. So this is a really just great image and set of figures from Josh Smith. This is kind of hot off the press, UC Santa Cruz researcher. So it's just really nice to see the kind of the timeline here. So kind of looking down at the sea stars, Pycnopodia timeline. So sea star wasting begins in 2013. We have a marine heat wave. These predatory sunflower stars, essentially dropping to zero, absolutely zero. They're still not seeing them anywhere in the California and Oregon area today. Purple urchin densities rapidly increasing, kelp densities declining, and then sea otter abundance increasing, all kind of within the same time frame. So they found that spatially explicit sea otter foraging enhanced the resistance of remnant forests to overgrazing, but it did not directly contribute to the resilience or recovery of the kelp forest. So they ended up with more of a patchy mosaic of kelp forests, and they're thinking that the otter behavior shifted kind of in response to the urchin densities and the urchin behavior, and so their outcome was tragic, but maybe not totally catastrophic. And I'll just point out too, in Washington, we do have our sea otters out in the Olympic Coast National Marine Sanctuary. A thriving population, I believe, is also growing. But one difference, we do have sunflower stars in Washington. The densities are much lower than they used to be, but we are seeing them in the shade of Fuca. The densities do decline as you go west out towards the ocean. So kind of moving to Northern California, this is the same story, but just a much more catastrophic outcome. Climate-driven shift in 2014, purple urchin population increasing by 60 times. Bull kelp canopy reduced by 90%, directly by the purple urchin overgrazing in just 1 year. It's just shocking. They also had a mass abalone mortality and fishery closure, red sea urchin fishery collapse, and right now they're dealing with action plans to reduce the urchin grazing pressure and enhance the kelp growth. So it's pretty tough times in that area. And you all might know Cynthia Catton or Roger Bennett as well. This is from their 2019 paper, and I just like the pictures say a lot about what happened. And I was just going to point out here, in Washington, shade Fuca, it still looks like this, okay, with that circle, kelp canopy, kind of everywhere you look, where you think it would be, but underwater, in some places, it looks like this, right? So it's not everywhere, it's not widespread, but it's patchy, and that's kind of what we're looking at right now. So it's just, that definitely has our attention. And I'll just kind of call out to pinto abalone in Washington are now endangered for the state. So we have a recovery plan in place dealing with pinto abalone, and obviously we have a red urchin fishery. So there's a lot at stake here, and this is kind of how it gets tied into our subtitle shellfish world. Okay, so kind of to deforestation by actively grazing purple sea urchin. The quick answer is hopefully not, right? And if it was, I think you probably would have heard about it by now. I think Helen would have already known, right? So, and then kind of second part, can we detect any signals of expanding purple urchin barrens and kelp decline? Yeah, we can try. So DNR, Helen and I and my colleagues that we huddled up and had many discussions of what we think we can do with maybe some existing data and what we can do going forward. Obviously, there's the aerial imagery of kelp canopy that goes back decades. And obviously, I think we're looking more in the last 5 years, so something like that. So yeah, we came up with a survey design, primarily with scientific diving methods here. So we established index stations, and the thought there was to be able to revisit the same exact spot over time, over basically once a year over 3 years. And we paired these stations as like predicted urchin barrens and a predicted reference. So those are fairly subjective. We kind of knew where to look for the purple urchins, so we would drop down, find an area that looked like it was impacted by purple urchins, created a site there, maybe about 400 meters away. It's pretty clear where you can find a spot where the kelp density is high and you're not really seeing many purple urchins, or none at all. We also had 2 depth bins, kind of like with the NOAA figure I showed before. So shallow 5 meter, deeper 10 meters deep, and the transects were 30 meters by 2 meters, parallel to shore. So the data we were collecting were urchin counts by species and kelp counts by species. That's 100% counts throughout the swath. Test measurements, test diameter of the urchins underwater, and we also collected whole live purple urchins for dissections to measure and sample the gonad index, or percent weight of the animal. We also brought a camera with lights to shoot photo quadrats. So that was half meter by half meter quadrat. Really hoping just to collect as much detail and resolution as we can while we're there. Didn't have very much time, and this was a big part of where Helen was able to come in. Her team already analyzed this a lot of underwater imagery, so this kind of fit right in. And just a quick example, you'll see in the upper right, this is a photo quadrat, just some of the highest kelp density that we saw, and below that you see some of the highest purple urchin densities in the quadrat, and clearly zero kelp anywhere. And then the dissections here, you'll see where you have this higher urchin density, almost zero kelp. You look inside, there's almost zero gonad in there. Okay, so starting more of a barren urchin signal. And up here, this is what it looks like when you have a full kind of gonad percentage of the body. And so, just for example, like this full yellow gonad inside that's what if you're harvesting urchins, that's what you're looking for. That's what you want. That's a high quality urchin. That's what sea otters would want if they were hunting as well. Moving on. So just a little more detail about the photo quadrats. Helen took this on, and she was able to get the macroalgae percent cover, kelp density by species. We also did urchin density by species. Another big part of what we could do with these photo quadrats was assess the grazing behavior. Just really challenging to do when you're diving and trying to do many other tasks. So we kind of looked at the percent exposed, and this comes from that Josh Smith study from UC Santa Cruz. We're looking at active grazing. So like an example, upper right picture here, you can see these purple urchins are out and about. They're exposed. Most of them are exposed. We call that active grazing. The top left picture here, good example of there's a pretty high density of purple urchins, but they're all tucked into their little spaces. They're not exposed. They do not appear to be actively grazing. So we use a threshold of 50% diameter or greater. We consider those exposed, actively grazing. So just kind of using that as another sort of metric and what we're looking at here. But some limitations of the photo quadrats. obviously the viewpoint can be kind of challenging, just the spaces within the rocks and how you can make some of these observations. The visible layers, obviously the kelp is laying on top of the bottom or laying on top of our kelp or obscuring other urchins. And then we're also looking more, to hold fast on the bottom. It's not really directly related to the canopy that's reached the surface. So,
Taylor, time to got a minute or two left.
Oh, okay. I'll just try to buzz through here. That's a lot. Yeah, generally, so here's a map to show you where the locations. I'm thinking of these more as like the east sites versus west sites, and then in the depth bins, the shallow and deep. But basically, yeah, the purple urchin densities and kelp densities were negatively correlated. So you can see, the higher where you saw the much higher urchin densities, the kelp densities are much lower. And kind of comparing the diver counts on the left to the quadratic counts on the right, so pretty similar trends there. These points are just an interesting explanation. They're kind of right in the middle. Those are actually these urchins are right on the edge of kind of a barren front moving into the kelp. So that's something we're going to look into a little bit more. And I was looking at the gonad index. It's really interesting. So urchin density and gonad index were negatively correlated. So you can see, and that is about what we would expect. Same for kelp density and gonad index. So higher kelp density, you're going to have a higher gonad percentage or index there. And those linear regressions were both significant. And kind of put it all together. So urchin density, kelp density, and gonad percentage, you kind of see real quickly, is really what we would have expected to see, relatively lower gonad index for the higher urchin densities, and over here, when you have a higher kelp density, again, the gonad index is going to be a little bit higher. And we did have a significant difference with so the west sites out but near Nia Bay are predicted barrens, significantly lower gonad index, around 7%. The total opposite of that over in the east. So the reference where there was a lot of kelp, significantly higher gonad index up near 30%. So quite a difference there. And this kind of just shows us, I think, we're getting more of that purple urchin barren signal out in the west, those western sites out near Cape Flattery, and really not so much over on the east side, kind of in that Port Angeles area. Even though they're relatively high purple urchin densities, it's not, I don't think we're really seeing that signal. So here, but Helen, what about the aerial imagery and kelp canopy data?
Taylor, do you have my slides? Can
I
you
do.
forward and I will start talking? Excellent. Okay. Some of you have seen, so Taylor asked me to take a look at our long-term monitoring data. And so here's a quick provisional summary. And there's more coming. First of all, as we know, and this is a format for presenting the data that Kathy Pfister showed us a couple of months ago. The kelp canopies along the coast and the strait, they tend to be highly variable. Here on the y-axis, you see canopy area as a deviation from the long-term mean. Purple is Nereocystis, which is more variable. Black is Macrocystis, which is less variable. And then you can see that there are links to climate. You can see PDO warm periods in red and cool periods in blue. Basically, as we all know, there's more kelp when it's cooler and more nutrient rich. There is no trend in this data, and I would ask you to peek at 2019. Things have not gone down. So we don't have a major urchin crisis. The biggest event for us in the last decade was that mean marine heat wave around 2014, where we saw really low kelp numbers and then a rebound. So that's the big picture, is we have a lot of kelp. Let's zoom in, next slide. These are the areas that where Taylor and the WDF and W divers put their sites. And what you'll notice is on the top is the area in each one of these index areas. So Cape Flattery on the left, Wada Island in the middle, and Tongue Point to Observatory Point in the right. And what you'll notice if you gaze at the figures of area is that we haven't seen big losses in 2019 relative to earlier years. And so at a larger scale, we are not concerned about major kelp canopy losses. And if you look below, there's a hatch mark pattern over a heat map of where kelp canopies most often occur. And what you'll see is that 2019, the hatch mark is over the red areas, which suggests that in 2019, that canopy kelp tended to occur in its long-term core areas. 2020, next slide. We're working on this data now. We're really struggling with resource limitations right now and kind of, but the 2020 analysis is underway. And what we're seeing is we don't have numbers yet, so we'll get back to you on this, but along portions of the strait, there's lower density and decreases. Here's one spot that I was particularly struck by. Here's McCurdy Point in 2019 and 2020. This is one of the largest contractions that really struck us. You can see that that bed to the eastern side has really contracted. McCurdy Point, by the way, is over towards Port Townsend in the DNR Reserve and Aquatic Reserve. Next slide. What we're mostly seeing, here's Wada Island, which Taylor talked about, 2019 versus 2020, is you can see slight, if you really look at these photos really closely, you'll see that that there's lower density and decreases in 2020. It's not going to be as much of a decline as 2014. And what we're doing now is finalizing this data in the next couple of months so that we can identify particular spots. Clearly, there are places where the urchins are clearing out kelp and other algae, but I think it's happening generally on the hundreds of meters scale from what I've seen. That's it.
Yeah, so just to kind of wrap up, the next steps here, more collaborations are on the way. We've got the Macaw tribe very interested in willing to participate as much as they can. More DNR, NOAA, Seattle Aquarium. It also has a presence out in the Western Strait. Curious about maybe the photoquadratic expansion, like is something that we can replicate more of, or maybe get other dive groups a chance to just expand that and more data. Another question: what's happening with the sea otter behavior? Is there anything we can detect there, similar to Central California? And then our plan now, we started 2020. We'll return there this year and 2022, and basically kind of see what happens over that 3-year time series. And hopefully everything is just kind of stable and we don't need to worry. And yep, thank you. A big shout out to the Macaw tribe and also U.S. Coast Guard Station Neah Bay for allowing us access to that area in 2020. I'm really surprised that we were able to pull that off.
So thank you so much, Taylor. And as with past talks, would everyone please add their comments to the chat, and Taylor can answer questions there. And we will move on to an update from Kelly Andrews at NOAA. They have sites along the outer coast moving into Neah Bay, and they have some preliminary observation on otters. So Taylor, you can stop sharing your screen. Perfect.
Can you hear me okay, Helen?
Yes, you sound great. I can't see, your screen is starting to show. I can see myself on your screen. That's weird.
I do not have anything to say about otters. Can you see my screen now?
Yes, looks great. Thank you.
Okay, cool. Well, thanks for having us here. I think as others have already mentioned, we have been doing kelp forest sampling out on the outer coast throughout the Olympic Coast National Marine Sanctuary for about the last 5 or 6 years, minus last year. And so this is going to be, just sort of a really short update on associations we've seen between kelp and urchins over the last couple of years. So first, this is mostly a collaboration between the Northwest Fisheries Science Center and the Olympic Coast National Marine Sanctuary, where we go out together to 5 different sites, which you see in red on this map here over the last 5 to 6 years. The first year we did this, we modeled this after some older scuba surveys that were done by Rick Kavitek. And you see those other sites as the black dots. So we did 10 sites that first year, and basically due to personnel, we just couldn't keep that up. So we've contracted down to these 5 main core sites, from Destruction Island in the south all the way up to Tatoosh and Nia Bay in the north. And so currently we have good data from 2016 to 2019, and then we basically intend to continue working at these sites until we're older and grayer, or we all retire. So and I guess I'll sort of just share what we do first. At each of those 5 core sites, we sort of break it into 2 different areas. Usually it's a north and a south area. And then within each of those areas, we run 3 to 5 30-meter by 2-meter transects at both the 5-meter depth and the 10-meter depth. And on each of those individual transects, we basically have 2 diver pairs. The first diver pair is taking data on fish and substrate characteristics. And then the second pair is collecting data on kelp densities and invertebrate densities, basically anything over 2 centimeters in length or radius. So that is the data that we collect, and I am going to keep this pretty short. So what I'm showing you here is our most recent calculations on density of kelps. We basically we identify every kelp species that we can. What I show here is Nereocystis in purple, and then Macrocystis in blue, Pterogophora in green, and all other species in yellow. And this is across each of the 5 sites. And then each row is at either the 5-meter depth on top or the 10-meter depth on the bottom. And probably the most, I guess, relevant patterns to focus on here for this discussion, and considering what Taylor just showed, is sort of focusing on on Tatoosh, where at the 5-meter depth here we've been seeing this decline in relative Nereocystis densities from 16 to 17 to 18, but in 2019 we sort of saw this rebound, although there's lots of variability and error around that estimate. And you'll see throughout across the sites, lots of variability in the ratio between the species. We really see lots of Macrocystis down at Cape Johnson and Nia Bay, and then Nereocystis as the primary kelp at these other sites. And to contrast that sort of change in direction of kelp density at Tatoosh, at the 5-meter depth zone, at the 10-meter depth zone, we've continued to sort of see this decline through 2019. Now, if we focus on urchins, such as the topic of this little period of time, we basically only see urchins at Destruction Island and Tatoosh, and a little bit at our Nia Bay site. But for purple sea urchins in purple at Tatoosh in particular, we definitely saw that significant increase up through 2017 and 2018, and then 2019 we actually saw a decrease, and that's observed at both the 5 meter and the 10 meter depth. Red urchins are significantly lower in densities at this site, and these are just a couple pictures that sort of gives you an idea of the pterogophora fronds that are completely eaten away in some of these areas, along with apparently urchins also like to eat bird eggs. So that was a really cool find. And then if you sort of put both of these together, what we've done here, and again, this is just real preliminary estimates here. This is showing kelp densities along the y-axis and urchin densities along the x-axis for each of the sites, which are are colored, and each of the years is what those numbers represent. So a 9 represents 2019, a 6 represents 2016. And if you look at, let's just focus on Nereocystis, that's the second column, and the purple urchins, which is the top row. Sort of the most significant thing that sort of popped out of here thus far is this negative relationship. Similar to what Taylor just showed us, also this negative relationship between kelp density and urchin density across this. This includes both the 5 meter and the 10 meter depths. But we don't see that at any of the other locations. So I'm just going to stop there and stop sharing.
Thank you so much, Kelly. And we will now hear from, so that was a great update on the outer coast and coming around to Nia Bay. We're now going to hear from Steve Rubin about the Elwha area, and then we'll have a little time for discussion. And keep adding questions to chat.
Hello, everyone. Let's see, I'm going to share my screen here, I hope. Okay. Go here. There. Okay, this is going to be another tag team presentation of 2 efforts that share the same study area. And here is the study area. It's Central Strait of Juan de Fuca. It goes from Crescent Bay on the west to Greenpoint on the east, and includes in the center of it near the mouth of the Elwha River. And this work started, at least for the dive surveys, which I'm going to talk about, because of the dam removals on the Elwha River, which happened a few years ago. But we've kept going, become kind of a long-term monitoring thing. And okay, so the black dots are the dive sites, and I'm going to talk about that in a few minutes. And the red lines are underwater towed video, which Melissa Sanchez from DNR is going to talk about. Melissa, are you ready?
Yes, can you hear me?
Yes.
Great. So you can go on to the next slide.
Oh, yes, okay.
Yeah, so for the past few months, I've been watching the towed underwater video of these transects that Steve showed in the red, and for the same Elwha study area. And I've been working on classifying mainly the sea stars, but also other inverts like the urchin populations. And so for these transects, there are video surveys that were completed for several years between 2010 and 2017. And then as well as last year for 2020, we were able to go back out and get video surveys for those transects as well. But most recently, I watched the 2012 and the 2020 surveys pretty much back to back. And one thing I noticed in this site in particular in Crescent Bay, which is the site which was furthermost to the west, I I noticed that I was seeing a lot more urchins in the year 2020 than what I had seen for 2012. And so the map here of Crescent Bay shows the survey transects broken up into 200-meter long sections. And I've used those sections to kind of summarize the areas in which urchins were either absent, present, or present in high density for the 2020 surveys. And then you can see in the deep edges that some of the transects not only had the urchins present, but they were present at the higher densities, similar to what you see on the photos on the left. And the 2020 surveys also showed urchins in these sections, but not at these high densities. The urchins in 2012 were more scattered and not as common or dominant. But in 2020, the urchins were seen in depths between -28 and -62 feet. But the higher aggregations didn't start till around the -40 zone and deeper. And see if you can pull up that video.
Yes.
I wanted to show a clip of what one of these deeper edges with the higher urchin densities looked like. You can go ahead and
Yep.
click play there.
Let's see here. Go like that and like that.
Great. And we don't have to watch it all, but it kind of gives you a better idea of what the underwater video is capturing and why these higher densities stood out to me. It takes a long time to go through this footage and classify the urchins second by second, like I'm able to do with the sea stars, which is why this analysis is just preliminary until I can go back and take a closer look at the urchins throughout the rest of the survey years and track a little more closely how big of an increase in urchins the video surveys are capturing. Yeah, but then we'll move on to the next slide.
Let's see.
And the next slide is showing the other 5 sites of the study area. And the presence of urchins at those sites. And the green shading was a little harder to see in these zoomed out maps, but I placed the little green circles around the sections where the urchins were present. And urchins were present in 3 of these sites here, Dungeness Bay, Dungeness Spit, and Freshwater Bay. And they were on the deeper edges of those transects, just like in Crescent Bay. But unlike Crescent Bay, the urchins here did not occur at those high densities like we saw in the video and the other photos. They were more like small groups that were huddled around the big rocks, or a few solitary urchins here and there throughout those sections. But my hope is to go back through all those surveys, probably with a focus on Crescent Bay, to get a more solid analysis and track the urchin populations a little more closely. But with that, I'll pass it back to Steve, who has more data on those urchins.
Okay, let's see, why is this not? There we go. Come on. Okay, so back to the map. So now I'm going to talk about the dive surveys, and they are the black dots, and just point out that we have a bunch of sites near the Elwha River, and then 2 farther east at Greenpoint, and so keep that in mind. And here's a blow up of the sites near the Elwha. Okay, so I should say more about we do the surveys annually in summer, July and August. 230-meter transects per site, PSCO method, offshoot. Our depth range is 6 meters to 18 meters. And this slide shows a big picture. This is just the average across all of our sites, 15 sites. including the ones at Greenpoint. And so the other thing about these sites are we have markers on the bottom, so we can go back to those. And so we're doing the same transects each year. And we started at these sites in 2011, and dam removal started right after that. And then sea star wasting hit between our 213 and 214 surveys, and it basically knocked out all the Pycnopodia at our sites. They're starting to come back, but they've been small, mostly small in recent years, the ones that are there. And then it took 3 years to take the dams out, and so that finished here. And the main thing about the dam removal was this huge amount of sediment came from the reservoirs behind the dams out into the straits, and it some deposits near the river mouth, but wider spread, high suspended sediment concentrations in the water column. All right, so urchins. This shows it broken down by red and green. We've never seen a purple urchin on our transects ever at any site. And so this is kind of the main, what I'm seeing here is there are more urchins now in recent years than there were when we started. Okay. Let's see. Now I'm going to break this down by site. 15 sites. 9 of the 15 sites have never had an urchin on them. This shows the other 6 sites. 3 of those sites had very low densities of urchins. This is density on the y-axis, and basically trace. So we're really talking about all the urchins at our dive sites were concentrated at 3 sites. And this, by the way, this shows the red urchin plus green urchin, the total urchins. And each line is a site. So we're just really talking about these 3 sites. And they reach respectable densities up, up over 2 urchins per square meter at this site. And I just want to, so here's one of those 3 sites. It's in the depth range that Melissa was talking about, where she also saw the high densities of urchins. And the other 2 are not on this figure. They're over at Greenpoint. There's both Greenpoint sites had high densities of urchins, and that was it. That's where the urchins were. Now these red dots show these are sites that we surveyed in 2008 and 2009. They're obviously different from the blue dots, and they're different each of those 2 years. This shows for both years, but so I thought we could go back in time and look at what those look like. So here are those sites. This part over here is exactly what you were looking at before, and these are for these random sites, and there were 29, about 30 sites in each year. Okay, so I was actually the way we did this is we randomized by substrate type, and so we had rocky reef and sandy and intermediate, 3 types, and we sampled equally in all 3 of those, but the distribution of those 3 substrate types were not equal. There was more. The least available of those habitat types was the rocky reef in our study area. So by sampling equally, we kind of high graded the rocky reef. So and that's that's something that at least I think urchins would like. So I kind of expected, I kind of expected these to be, if there were equal numbers of urchins here in this time period to here, I kind of would have expected these to be higher. So I think this kind of this suggests that this supports that there have been more urchins recently than there were before. Yeah.
Steve, go ahead and wrap up, please.
Okay.
We're out of time.
All right. This is big picture kelp, and I'll just point out that the kelp in the study area, this is average over all the sites, and the kelp are doing fine in recent years. Kelp of all species are doing fine. That's it.
Thank you so much, Steve, Kelly, and Taylor. We're going to keep moving on because we're out of time for the urchin topic, and we want to wrap up on time. So for the last discussion topic, we've asked Betsy Peabody to lead a discussion of new projects that were funded in the recently approved state budget. Are you here, Betsy?
Here.
I saw you earlier. There you are. I can hear you.
Here. Okay, thank you, Helen. And many people on this call actually participated in this, but the stars sort of unexpectedly aligned, and we were able to get some kelp funding from the legislature this session. And we got 1.493 million to fund the work by 6 entities around Puget Sound. And those include Northwest Straits Commission, Samish Indian Tribe, Lower Elwha Clallam Tribe, Friday Harbor Labs, Puget Sound Partnership, and Puget Sound Restoration Fund, and all engaged in diverse actions that were identified as priorities in the 2020 Puget Sound Kelp Conservation and Recovery Plan, spanning understanding stressors, monitoring distribution and trends, and also testing the efficacy of seed outplants to bolster populations. Those were sort of the main actions funded. But I thought it actually might be really interesting to do kind of an analysis of the critical factors that enabled this funding. And I want to begin by just saying that this call demonstrates the incredible work across the Salish Sea that's going on by many diverse partners. And so there's sort of a critical mass of attention on the importance of kelp forests. And I think that that's one of the major factors for why we were able to get funding. And also there have been really wonderful investors, early investors in this. So NOAA providing funding, all the work that DNR has done, all the work that many organizations on this call have been doing. So a lot of commitments by many partners to doing kelp work. And what happens when you have that critical mass is that the word about kelp forests starts to trickle up to legislators. Okay, they're hearing about kelp from lots of different people with whom they're having conversation. So it's getting their attention. And that creates an opening for a legislator to invite a kelp request, as happened in this particular case. So Senator Rolfes was hearing from a number of us about kelp forests and the need to really move forward in a big way with conservation and recovery efforts, because this is such a critical part of the health of Puget Sound. So she's hearing this, and somehow or other there's budget at the state level, unexpectedly, right? Coming out of the pandemic. So that creates an opening, and she invites that request. But then actually, in order to navigate this particular thing, the way that you have to thread this needle is you have to put together an ask that is a consensus decision by all the different partners involved in Kelp in this area. So we've got to reach consensus over which tasks are important, right? And we have to be proposing something that the legislators want to fund. So there have to be diverse actions by diverse partners in order to get that kind of support from the legislature. So that's kind of how we were working this through the process. And then there have to be state agencies who are willing to funnel that money and administer that new funding to support this work over the next 2 years. So that's another critical factor that has to fall into place. And I will just say that Dana, if you're on this call, Dana Oster with Northwest Straits Commission has been such a great partner in this, in terms of both sort of, you know, identifying those priority tasks and helping to craft that language so that we had something that we could present to the legislators. And so really appreciate that partnership. And then also Northwest Straits Commission was willing to be one of the state agency partners that would help administer that funding, along with Puget Sound Partnership and University of Washington as the administrator for the Friday Harbor Labs funding. So those are all the things that have to fall into place, and that did magically fall into place this time around as a result of many people on this call working this through their different angles, and also as a result of Northwest Indian Fisheries Commission declaring it as a priority and urging legislators to fund the full funding request. So that's kind of an anatomy of how this happened. I think that the next steps is for us to embed this work and continue to drive this work by including funding in state agency budgets that can keep these various partnerships alive and keep us all working on these priority actions. So that's kind of a next step. And I would say, just as one more thing, just why it's important for us all to be talking to our legislators. Because again, when we want them to know what's happening in these waters, all the incredibly important work that everybody's working on. And as they hear about it from all these different players, then again, they start to pay closer attention, and it helps provide funding support. Because for us to do this work moving forward, and there's a lot of really important work, we are going to need to continue to get that kind of state support and federal support and funding through research agencies and NGOs and foundations, et cetera. So shout out to everybody for help, for your help in making this happen. Obviously, the 2020 Conservation of Kelp Plan was critical, just in terms of creating that roadmap, and then being able to say, hey, we're implementing that plan and we need funding to do that. So thank you. And I'm glad that we'll all be working on different things with this state funding starting on July 1st of this year and continuing through June 30 of 2023. That's it.
Are there any other comments that anyone in the funded groups would like to make? We have 2 minutes, 2 full minutes left in this meeting. And we would love to hear from either someone who wants to make any further comments on the funding or on any other priorities that they would like this work group to have. And you could either speak or you could add it to the chat.
Hey, I had a question actually. I know this came up in the PSEP meeting yesterday, I think. Is there a link to a document that shows kind of the breakdown of that funding approval somewhere that people can check out?
Hey, Dana, are you on the call by chance? And if you're on the call, I wonder if you can include that link in the chat.
We haven't put it in the link yet. We have a PDF and I can try and upload it as a file or just give to Helen, maybe if it seems appropriate for the website. But I'll work on that now.
I can put it on the website. I can put it on the website.
This
will
is
do that.
Natalie. I have a quick link to it that I'm happy to paste in the chat.
Thank you, Natalie.
Thank you.
So on that note, it's noon. And so I'm going to make a last call for any comments, either verbally or into the chat for about today or about things we'd like to do in the future. Please let us know. And by the way, thank you so much. We have a really great and large group here today, and it's just really exciting to get to hear about all this good work that's going on. And thank you so much, Betsy and Dana, for all the work you've done. Okay, I think we are done. Thank you so much, everyone. And I'll hang around for 5 or 10 minutes if anyone else wants to speak. But this meeting is officially over. And if you have anything to share about upcoming projects or results that you'd like to share at this meeting or distribute to the group, let us know. Thank you, everyone. And especially thank you all to the presenters. It's great to share this information.
Thanks, Helen.
Okay, I just put the link to the legislative request PDF in the chat. Should be publicly available to everybody. This is Natalie. And
thank you, Natalie. I see it. That's really helpful.
Okay.
And we will also, we've started building a little news bulletin that comes out of this, and we'll make sure it's in there too.
Thanks, Helen. This is Todd.
Hey, Todd.
How's it going?
It's going great.
Good. I was excited to hear about some of the, dive survey protocol and folks using ROVs. That's part of what we've been funded for in the legislative request, as well as a BIA grant that we received. So I'd be interested in connecting with some of those folks so that we could do comparable methodology with our ROV and with our dive teams. As my first, I have one person who's already certified, another one's in class right now, and the third one starts class next month. So we're going to have some added capacity up here in the San Juans, but would like to make sure that we're developing protocols in-house that match what other folks are doing so that we can compare data better. So
Todd, I really appreciate you reminding me of this, because it feels like we're getting a critical mass of people who are interested in diving. And so I'm going to send out, including the DNR divers may be able to pick up some index sites down here, the Washington DNR divers. So I'm going to link you into that discussion. Have you already heard from Dan Abbott, who's hoping to implement Reef Check? So that would be a volunteer dive group?
Have not. So
okay.
So we're coming out with our initial, just open water cert. So we're not scientific divers yet. We're certainly going to be looking at added qualifications and certifications that we can add on in the future. But so right now, I think we'd be most comparable with sort of those citizen divers, except that we have really good landing craft that are amazing platforms for dive support. So we'd be interested in partnering up with folks here in the San Juans to be, force multipliers or whatever. So.
Yeah, it's I'll send out an email following up, and anyone else who's still on the call can weigh in. But two people who are key are Steve Rubin and Matt Byrne, who are still on the call. And then also Eliza's new work, because and then the PSRF work. But yeah, I'll send out one email for connecting then, and also get you hooked up with Dan Abbott.
Yeah, I have been in contact with folks at Puget Sound Restoration Fund, so we're having some conversations as well. But yeah, I think it's just it's turning out that we're adding more and more work in the San Juans, and I know there's interest from the entire group in filling data gaps and working up here as well. So we want to help for sure. So, and now we've got some funding to be helpful. So.
the funding has been amazing. It really makes me optimistic and hopeful.
I actually have another grant to U.S. Fish and Wildlife out that we're still waiting on, and in which case I'm going to have funding for kelp that I'm not going to be sure what to do with. I'm going to have to, I don't have enough people or boats at this point if I land all these grants. So yeah, the legislative request was an exciting boost. And what it does is sort of double our ability to look at sites compared to what we were funded for, prior to that. So pretty exciting.
So I see some other people hanging on the call. Does anyone else have anything? I could ask Todd a million questions, but before I keep talking, does anyone else have other things that they'd like to raise to the remaining group?
Helen, I can chime in. This is Natalia. I was didn't chime in right away when you first asked, just trying to leave some space for others to talk about their requests. But for those that are still in the call, I'm Natalie from the partnership, and I manage our indicators program, the vital signs program, and we did get a proviso to help develop a kelp indicator for Puget Sound. So, everything we heard this morning and what Tal was just talking about too is relevant as we are looking to use these resources to support a collaborative project and a project lead that has the expertise to do things like inventory data sets and test protocols that could be implemented by partners and integrate and synthesize data to produce a region-wide indicator. So this was good news for us since we didn't have, we're always short on resources for our indicators at the partnership. We have quite a few of them. And I wanted to add that our first step here is to take advantage of the current monitoring solicitation that's out in the streets right now, looking for project proposals. Kelp indicators are one of the priority information needs in that solicitation. The proviso aligns well with the scope of the projects that we're looking for. So the letters of intent are due on Friday for the solicitation. So from the letters, we'll see what projects come in, and from there we'll evaluate our options for moving forward with this funding. So I just wanted to connect kind of the proviso work to the monitoring solicitation that we have out for you all. And I am absolutely sure that we will keep this work group updated and maybe even involved for this project, for the indicator project. So thank you.
And
so Natalie, oh, go ahead, Todd.
We should also chat because the bulk of what we've been funded for over the last few years is digitizing aerial photography. So we can actually run calculations in Samish traditional territory of approximate kelp canopy coverage, which seems to ally pretty well with the initial indicator that is on there. So yes. You can check out our story map linking to Samish's website if you'd like.
I would love to.
But we should chat about how to share, the raw data or whatever information is useful to the partnership to actually be able to, provide some immediate data for kelp canopy coverage for that indicator up in the San Juans.
That sounds great. Would love to. Helen, did you have something too? Or do you want to move on to something?
I had a question that was asked of me, and then I realized that I had the same question myself, which is, this is a very detailed question. For the letter of intent, should the budget be part of the 3-page total? And is it just a very simple outline of a budget, or is the budget separate and should it be detailed to the detail of Appendix D?
Thanks. So the budget is part of the 3-page, and you can, we would like a budget breakdown. So definitely look at how we talk about that for the letter of intent, not for the full proposal. And it's really just to help us understand, what's all involved, who's involved, and what kind of resources are needed, and to help us sort of better assess kind of the potential for success of this project. So we do ask for some level of breakdown, but to the extent that you can. And if you look at the Q&A document, there's a question and answer related to budget in there too that should reflect what I just said, but definitely go check that out. But yes, part of the 3-page.
Got it. Thank you.
Single space.
Single
Single
space.
space. So, yeah.
Can you use a 2.5 font on that? Or?
Yeah. Don't make us strain eyes too much.
Okay, if there are no other comments, we can officially end this meeting. Does anyone want to add anything else?
I'll quickly add, I think, the link to the monitoring solicitation in the chat.
That would be great. Okay.
Thanks,
Have a good
everyone.
day, everyone. Thank you all for joining. I can't tell you how exciting it is to see. We had a lot of people here today.
Yeah.
It was great.
Thank you, Helen.
Thanks, Hugo. Bye,
Thank you.
Steve. Bye, Matt. Bye, Betty. Bye, Natalie.